All right. Well, my name is Doug Dean, and I'm a postdoc research fellow here at the Waisman Center. And I'm really excited and it's a real great privilege to be here and be able to share some of the research that I'm conducting with here at the Waisman Center, looking at quantitative magnetic resonance imaging in brain microstructure within individuals with autism spectrum disorders. And before I actually get started with describing to you what we actually do, I want to first acknowledge what a great team of researchers and collaborators and colleagues that I get to work with every day, not only being able to come here at the Waisman Center, which is an amazing place, as you just heard from Chung, but also working with such a great, wonderful group of people, really spearheaded by Drs. Andy Alexander, Janet Lanehart, who some of you might have seen passing through, participating in research here. Really, it's a wonderful group, and I can't really acknowledge or be thankful so much. And really, our research program for our lab is really to develop and apply advanced brain imaging techniques to, one, determine changes in the brain specific to autism and that might be linked to specific symptoms or outcomes that we see in individuals with ASD, but also to use this type of research to identify brain-based biomarkers that may inform on different causes of autism, different neural mechanisms that might be involved within the symptoms that we see, aid in early diagnosis, and also try to develop preventative interventions and or treatments for ASD. So we use a lot of different techniques to study the research participants that come in through the lab, but I'm really going to highlight some of the methods in that we use for brain imaging. And so to give you just a quick analogy for those who might not be as familiar with magnetic resonance imaging or brain imaging in general, so you might be more familiar with the idea of taking a picture with a camera. So we have a picture with a camera, or you can take a picture with a camera, and you get this really nice photograph of, for instance, my twins back at home. And so in brain imaging, our camera is actually this big magnet. So you lay down in here, and it takes a little bit longer than taking a photo with your camera, but after about 10 minutes or so, we can get these really nice, clear, detailed pictures of the brain. And what's really interesting about MRI is that we can use different types of cameras to get different views of the brain, but also we get this 3D information. It's not just that we get this single picture of the brain. We can look at the brain in all different kind of views and aspects. And that's really exciting because we can use that to look at different parts of the brain, but also ask questions just about the whole brain itself. And one of the reasons why magnetic resonance imaging is so popular in medicine specifically is that it gives us these really detailed, clear pictures of the underlying brain tissue. And in different clinical pathologies, say for like MS or traumatic brain injury, it's really clear to see where something might be wrong in the brain. So in this particular case, you can clearly denote this lesion that exists, and for a doctor that's very informative. But if we were to look at two participants, one that has autism and one who may be typically developing control, those differences are are much more subtle, or at least not visible with some of these traditional imaging methods. And if I were to ask a clinic or a doctor who's really trained at looking at these images, which one of these participants has autism, they would not be able to guess correctly, or maybe guess by chance 50% of the time. And indeed, based on the diagnosis through different behavioral diagnoses, and this one on your left is one that we see with autism. But again, there's no clear, visible demarcation in the brain that tells us that this is the case. Well, I'm going to go back and refer back to this idea of taking a picture now. And just like you can do on your phone, for instance, or we can apply different lenses to a camera to get these different pictures, we can do the same thing with MRI. So we can, what we call, instead of a lens, we call a sequence. So we can apply different sequences or use different sequences from the MRI scanner to get different types of information from the brain. So for instance, we can get images that relate to brain structure or anatomy. We can also look at play different sequences that give us information about brain function, and also different more advanced sequences that give us information about brain microstructure. And that's going to be what I'm going to really focus on today during the rest of this talk, and describe how we're using advanced techniques to really look at the underlying microstructure in the brain. And one such technique is, you might, those who are familiar with imaging might have heard, is called diffusion tensor imaging. And really this technique probes the tissue microstructure by investigating how water diffuses throughout the brain. And the nice thing about DTI, or diffusion tensor imaging, it provides these quantitative imaging measures that reflect the underlying microstructure. In particular, this fractional anisotropy, or FA, is kind of a ubiquitous measure that is used in many different studies, not just in autism, but a whole host of different pathologies in that. That gives us really clear information about the underlying white matter microstructure. And when I talk about white matter, what I'm really referring to is, if we think of our brain as a computer, we have basically two different types of tissue. One is the gray matter that looks kind of gray here in these pictures, and you can think of those parts of the brain really reflecting the processors of the computer. They're the ones that do all the thinking. Whereas the white matter that's shown here in white is actually all the different wires that connect those processors together. And those wires, without those wires, it'd be really difficult for these processors to communicate and also do what they need to do. And the same thing holds for the brain. So without this white matter being able to connect the different parts of the brain together, we're not able to function as proper, or as what we would normally be able to do. So if there, for instance, in multiple sclerosis, where this white matter is affected, you see a lot of different motor or behavioral issues or problems with such a pathology. And so we're really wanting to know this wiring in the brain is influenced in autism spectrum disorders. And we know for some previous research conducted here by the center, by some collaborators and colleagues, that white matter does matter in ASD. So being spearheaded by this longitudinal, 15-year longitudinal study that really started by Dr. Andy Alexander and Janet Lanehart, following individuals across the lifespan with autism spectrum disorder, as well as those who are typically developing controls. And what we find is that across that lifespan, we do see these developmental differences across with some of these different imaging measures that we see. So for instance, on the x-axis I have plotted here is age, and then on the y-axis is this fractional anisotropy from one of the more important white matter regions, the corpus callosum. And what we see is that early on in life, there might be individuals here in blue are those with autism spectrum disorder, and in black are those who are the typically developing controls. We see this kind of interesting pattern that, there's some different path or crosses here indicating that FA is increasing in the typically developing controls, but more or less staying maybe kind of plateauing or staying relatively flat across the lifespan, which again kind of is an interesting trajectory that gives us information about how the white matter is changing throughout the lifespan. More recently, in another cohort of individuals that we've looked at here at the Waisman Center, we've seen very similar trends. So again, we see early on in life that individuals with autism have a higher fractional anisotropy early on, but then over time that pattern changes, and then the typically developing controls eventually overcome or surpass that trajectory of the individual with autism. And then if we look at different other areas across the brain, like I said, with MRI, we can really look at how things are changing across the brain. What we see is similar patterns, not just in the corpus callosum, but in other brain structures as well. So in kind of the thalamic regions and the internal capsules as well, in parts of the brainstem. So again, this gives us nice insight into what areas of the brain might be influenced or affected when in individuals with autism. But when I show some of these things of these group differences, it's really important to realize that not every individual might be affected in the same way. There's a very clear and widespread heterogeneity across different individuals and across different white matter regions. For example, more recently we've been trying to identify ways to capture this individual heterogeneity, and we've come up with some cutting edge measures to do so. So here on the x-axis, we're plotting a measure that relates to how far away an individual is from the normative growth curve. So you kind of think of it as when you go to the pediatrician, they give you for your child, they'll give you like a percentile of how far away they are along that curve. You can kind of think of this the same way for the brain. So we see in blue here the typically developing controls, and there you see this nice blue distribution that's really close to zero, indicating that they're closer to that normal control pattern. But then in red, you see all the individuals with autism, and you see some individuals here for this specific brain region do indeed look fairly normal or typically developing. But then there are other individuals who are a bit further away from that typically developing control growth curve. And the one thing to really note with the individuals with autism is in that we see this distribution have a lot more variation. So there's this heterogeneity across different white matter regions, and those individual differences really depend on different brain regions. So for instance, this particular brain region here that we're looking at, for the most part, our individuals with autism look very much like those who are typically developing, suggesting that not every brain region is affected in the same way. When we start to tally up different white matter measures across different brain structures, we then begin to be able to separate out those who are typically developing from those who have autism spectrum disorder. So by combining different brain regions across the brain, we're able to separate out and really show that it's not just a single brain region or structure that is influenced in autism, but it's a combination of different measures. And by doing so, this provides greater sensitivity for identifying those individuals with autism rather than just looking at a single brain region. And one thing why this is really exciting is that this now allows us to construct, using this type of measure, a phenotype based on brain measures. And by doing so, we can then take those brain phenotypes and go back and work with psychologists or geneticists to look at underlying behaviors that might be associated with this phenotype, but also begin to probe and look at different genetic causes or environmental differences that also might relate to these differences. And so while I've described diffusion tensor imaging and being this really nice imaging technique that provides detailed information, the one problem with it, one limitation of this technique, is that although it's really sensitive to that microstructure, it's very non-specific, meaning that there are different processes in the brain that could cause similar changes in these measures. For instance, changes in the way that the white matter fibers, those wires, are organized might lead to these increasing FA, but also other changes in the white matter can lead to similar changes. So we don't really, using DTI, we can't understand what processes are actually happening when we look at these measures. And so just as your phone gets updated every year or your computer gets updated, there's been an explosion of these advanced microstructural imaging techniques in our field where we try to improve our measurements and characterization of the white matter for understanding processes underlying the changes that specifically we're using and applying those, developing and applying these techniques at the neurodiversity in autism spectrum disorders. And one of these techniques that we're really spearheading and excited about is a technique that's called NAUDI, but it stands for Neurate Orientation Dispersion Density Imaging. And it's a unique imaging technique that provides measures associated with brain neurite microstructures, so kind of the axons and dendrites in the brain. And really, this technique is relatively new, and it has not been extensively examined in individuals with autism spectrum disorder. And so we've been, like I said, we've been spearheading and really excited for this technique, and for the first time, we're looking at some of these measures within our local cohort that we've been collecting for the last few years. And really what we're beginning to see is that in individuals with autism, we see this pattern of lower neurite density within the thalamus, which is a really interesting part of the brain supporting different sensory functions, which is very interesting in the recent result that we've seen. And if we look at that trajectory again across the lifespan, we see that kind of same pattern within this thalamic brain region that we see using some of the other measures that I've previously shown. We see that again early on in life, the individuals with autism seem to be actually a bit higher with this measure, but then they plateau a little faster than that of the typically developing group, where we then see kind of increases. And what we're actually really excited or interested for using these techniques for is that not only do we see these differences between these two groups, but we also see that this measure is highly correlated and associated with autism severity measured by the social responsive score. And this is, again, a really exciting finding that we're preparing to write up as currently. But not only do we see this lower neurite density, but we also see parts of the white matter that actually look more coherent, and there's less kind of dispersion in the brain. So this here in these areas here in blue are showing that. We see that in the internal capsules in the right superior longitudinal fasciculus, there are actually areas of white matter that seem to be even more coherent than that of our typically developing group. So we're trying to explore what some of these processes and measures might mean, and what they might tell us about the symptoms and outcomes in our individuals. Summary. So I've basically shown that white matter microstructure plays a very important role in the neurobiology of ASD. And despite the group level differences between ASD individuals and typically developing controls, there's wide variation within the brain that we can really use to probe different aspects of the disorder and try to look at how we might be able to tailor different interventions to treat some of these specific brain regions. And then also emerging techniques like NAUTI and others that are more recent provide new aspects for studying white matter, and they may inform on some of the underlying neural processes and the neurodiversity that we see in ASD. And really that's the key part for using brain imaging, is that we need to be able to understand what is that underlying microstructural process in order to be able to help develop new techniques and possible treatments in intervention. And so looking forward, interests are really looking at early brain development. So we know that the brain develops very rapidly, or child develops very rapidly across the first 2 years of life, for instance. But we also know from imaging kids very young in life that there's tremendous brain development during this time. So it's one of the fastest growing periods of development is the first 0 to 2 years of life. We see an explosion of white matter development in the brain. So early on in life, there's very little myelinated white matter, and over time, that process is very rapid. And by about 2 years of age, the brain is already looking adult-like. And so by understanding some of these processes early in life, that might be very helpful for looking at how a child who goes on to develop autism has developed prenatally. And also, based on early brain development, we know that this process of early brain development overlaps tremendously with different stages of behavior. Different brain networks are developing as these different behavioral processes are coming online. And so there's, we know that brain development plays this critical role in behavior, in the development of behavior and cognition. And so we want to understand how these processes are linked. And really, there's very little previous research looking at this critical time period in individuals with autism, more or less because it's difficult to diagnose individuals at an early age, but by following children and watching how they grow, we can maybe be able to begin to understand the neurodevelopmental trajectory of ASD early in life. And then can we use some of these new and emerging techniques for microstructural imaging to provide that kind of new information that we would really like to know. So with that, I will again acknowledge the wonderful team that I get every day, and really the sincerest gratitude to all the participants and families that participate and make our research possible, and also all the different funding resources that we have been so fortunate to have. Thank you. Yeah.
Can you say something about your sample and the issue of consent?
Sure. So our sample that we've collected here is a pretty widespread. So we have kids or individuals between 10 and 40 years of age. And usually the consent process is done in conjunction with family members. So we would have parents help with the process.
Here, I've got the microphone.
Is there some concern about subpopulation selection based on ability to tolerate the MRI?
Yeah, that is an excellent question. So that is one of the big limitations that we have when studying ASD, is that a lot of kids who have very severe forms of autism and may not be able to tolerate the scan, or if they are able to tolerate, they tend to move a lot in the scanner and kind of distort the images that we collect. So here at the Weisman Center, we're trying to overcome that by developing some new strategies for correcting for some of that motion. So if individuals are able to make their way into the scanner, we can hopefully correct for some of the motion artifacts that we might see. We also try to make the scanner environment a lot more friendly of a place for them. Sometimes we can dress up the scanner as like a rocket ship or something exciting that makes it a little less clinical for individuals, and that sometimes helps as well. But yeah, that is definitely a limitation of the research that we do. And generally, we don't use any types of sedation or anything like that for scanning, at least for research.
Any other questions?